So we initially discussed two stories. One is that of Sanger sequencing technique, and we'll talk of it in more details as we move along. And the second is how the Sanger sequencing was used to sequence the human genome. And this is where the need for large insert vectors arose. And we'll go through these stories before we actually come to the large insert vectors, that is bacterial artificial chromosomes and yeast artificial chromosomes. Here is a picture of Fred Sanger, a Nobel laureate, and not once but twice. And one of the Nobel Prizes that he got in 1980 was for developing the technology that is known as Sanger sequencing, for sequencing a piece of DNA. And the duo shown here are Craig Venter and Francis Crick, people responsible for the sequencing of the human genome. And we'll talk of these stories in parallel as we move along, and then finally move on to the actual features of large insert vectors, right? So I've already told you that Sanger got Nobel Prize twice, one of which was for developing the Sanger sequencing technique. The activity that I want you to do is to find out what is the other contribution for which Fred Sanger received a Nobel Prize way back in 1958. So let's now discuss the Sanger sequencing technique first. So DNA polymerase is one of the key enzymes required for replication of DNA. Also, when you do an in vitro amplification in PCR, you would require DNA polymerase, right? And DNA polymerase has certain prerequisites for amplification of DNA. One, you must have a template strand here, so which is shown here. This is a template strand. So second, what is important also is that DNA polymerase cannot extend a DNA sequence de novo. It requires a pre-existing oligo on which it can add complementary nucleotides based on what is present on the template here. So therefore, the second important requirement for DNA polymerase is a complementary oligo to the template. This is known as a primer, since it cannot synthesize de novo. So here you have a primer. This is, if you see here, the 5' end is present here, and the 3' end is where the extension of this primer is going to happen, and which nucleotide is going to be incorporated here would depend on what is the nucleotide on the template. A complementary nucleotide to this will be incorporated here. Third, of course, what is important is the presence of magnesium ion, which is required as a cofactor for DNA polymerase to function. So once you have this sort of arrangement where you have a single-stranded template and a complementary primer annealed to it, you can start your synthesis of the complementary strand here, and DNA polymerase, based on what is present on the template, would add a complementary nucleotide on the growing strand, right? So here you are, for adenine, it adds a thymine. That second nucleotide is a thymine, so it adds an adenine, cytosine, guanine, thymine, and so on and so forth, right? So depending on what is the length of the template that you have, you will have your extension happening here. So this is the basic function of DNA polymerase. So we could represent this reaction of addition of single nucleotides with this equation here. So here, this DNA whole N is the primer sequence here. This is represented as DNA whole N. And you're giving your ddNTPs to the reaction, or deoxy nucleotide triphosphate, in the presence of DNA polymerase. This DNA of length N would be extended by one nucleotide at a time. So you get DNA N+1. There will be a release of pyrophosphate and an H+ ion alongside in the reaction. Then it moves to the second nucleotide on the template and adds a complementary nucleotide on the growing strand again, and so on and so forth. The reaction is continued, right? So this is the basic reaction that happens in DNA polymerase. So if you look at the actual reaction that is happening here on the strand that is getting extended, so every time a nucleotide is incorporated, what you are doing is actually forming a phosphodiester bond between the 3' OH of the previous nucleotide and the 5' phosphate of the incoming nucleotide. So this would have a 3' OH, the incoming nucleotide would have a 5' phosphate, and the 3' OH and the 5' phosphate combined together, removing a water molecule and forming a phosphodiester bond, right? So what is happening in this reaction actually is the formation of a phosphodiester bond. Let's say we're talking of this position here. The phosphodiester bond will form between the 3' OH of the pre-existing nucleotide on this strand and the 5' phosphate of the incoming nucleotide here, right? So therefore, for the formation of a phosphodiester bond, there must be a 3' OH group present on the 3' end of the strand where the addition is going to happen. and the incoming nucleotide must have a 5' phosphate group. So what Sanger very smartly did was to introduce nucleotides that did not have a 3' OH end. And therefore, once such nucleotides are incorporated, no further extension of this fragment was possible, because for formation of a phosphodiester bond, you need a 3' OH and a 5' phosphate of the incoming nucleotide. Right? There is a slight modification in Sanger sequencing of this process that allows you to generate fragments that differ from each other by one nucleotide. So what Sanger very smartly did in his technique for sequencing the DNA was to introduce, along with normal ddNTPs, dideoxynucleotide triphosphate. As the name indicates, dideoxy means at two positions there would be no OH group. So it is not there at second carbon on the pentose sugar, and the OH group is also absent at the third carbon on the pentose sugar. And because there is no 3' OH, once a ddNTP nucleotide is incorporated, the fragment cannot be extended any further, because no further phosphodiester bond formation can happen. For the formation of a phosphodiester bond, you require a 3' OH at the last nucleotide in the oligo and a 5' phosphate in the incoming nucleotide. Since the 3' OH is absent, you cannot have any further extension of this fragment, and that forms the crux or the basis of Sanger sequencing. So let's look at this reaction in more details. So here you are, this is a template, and you would be synthesizing a complementary DNA strand to this template, which will basically be extended on a primer here, right? So here you are, this is the primer. The 5' end, 3' end is where the new nucleotides will be added. And while you're adding the new nucleotides, you have a choice between adding a complementary dNTP or a complementary ddNTP, right? If you add a complementary ddNTP, then there can be no further extension of this fragment here. Why? Because there is no free 3' OH group on this thymine. You cannot have formation of a phosphodiester bond with the next incoming nucleotide. So therefore, now this fragment is frozen and cannot be extended any further. However, because we've also given normal dNTPs in the reactions, there is a chance of adding a normal dNTP, in which case the polymerase would now come to the second position, look at the template and see what is the nucleotide here, and add a complementary nucleotide at the second position. Again, at the second position, you have a choice between a dNTP and a ddNTP. If a ddNTP gets added, then again, no further extension is possible here. Likewise, if a normal dNTP is added, then you move to the third position. There is a guanine on the third position of the template. You can now add a dNTP, complementary dCTP or ddCTP cytosine here, right? So in case you add a ddCTP, again, this fragment cannot be extended any further. However, given that we have dNTPs and ddNTPs in a certain ratio, some of the fragments would incorporate a normal cytosine. So in which case it moves to the fourth position here, which is a cytosine on the template. So a guanine will be added here. Again, a dNTP or a ddNTP. If it is a ddNTP, no further extension of this happens on this fragment. However, in case you have a normal guanine getting incorporated, then you come to the next position again, and again you have a choice between DTTP or ddTTP thymine, right? So here you are, you have your incorporation of dideoxythymine here, and because it is dideoxythymine, no further extension of this fragment can happen. However, in alternate case where you have a normal thymine getting incorporated, you move to the next position, and you can again have a dideoxythymine incorporated or a normal thymine getting incorporated here, right? Or deoxythymine getting incorporated here. So this is the way the Sanger sequencing proceeds, and what you effectively get after you've done your reaction here is fragments that differ from each other by one nucleotide. And if you can see, I've given a different color to each of these ddNTPs, and this is what actually happens in automated Sanger sequencing. Each of the 4 ddNTPs are differentially labeled with the fluorochrome. Each has a different color, and that allows us to detect which nucleotide is present at the 3' end of a given fragment here, right? So very clearly, if you can see here, what we generate is fragments that differ from each other by one nucleotide, and each fragment actually ends in a dideoxynucleotide. So these represent the collection of fragments that you obtain upon extension by DNA polymerase and incorporation of DNTP or ddNTPs, right? So once your sequencing reaction is over, the next step here is to basically separate these fragments on the basis of size using electrophoresis. And in automated Sanger sequencing, what you use is capillary electrophoresis, right? To conclude this part of the talk, as an activity, I ask you this question. DNTPs and ddNTPs differ from each other with respect to which carbon atom on the pentose sugar. So as I said, the next step here is to size separate these fragments using capillary electrophoresis. So here you are, you set up your capillary electrophoresis. This is a plus end, this is a minus end here. You know DNA is negatively charged and therefore it would move towards the positive end. And upon electrophoresis, the shortest fragment would run the fastest and the heavier fragment would run slightly slower. So when you separate out these collection of fragments that you obtain using DNA polymerase and ddNTPs and dNTPs, they would separate out on the gel in this manner here. So you have the shortest fragment coming up the fastest towards the plus end, while the largest fragments would basically be at the top. So here you are, and you can see each of these fragments is basically having a fluorescent ddNTP, and that allows us to detect which nucleotide is present at each of these fragments. So what you do is, in automated Sanger sequencing, there is a laser that would detect the color emitted by the fragment, and that allows us also to translate it back to the actual nucleotide sequence, right? So the fragments are resolved on gel, and there is a laser that is used to detect the intensity of color being emitted from the ddNTP that is present at the 3' end of each fragment, and that allows you to translate it into the actual nucleotide sequence here. So this is what is the basis of Sanger sequencing. However, there are limitations to the technique. So one of the major limitations of Sanger sequencing is the fact that you could only sequence around 900 bases starting from one end, right? So now the question is, if you have a larger piece of DNA to sequence, how do you go about sequencing it? So one obvious solution to the problem is to break the larger problem into smaller individual problems. So what we do is we break this long piece of DNA into smaller fragments that are amenable to Sanger sequencing, generate the sequence for individual fragments, and then rearrange these fragments back in the order in which they appear in the actual larger DNA here, right? So finally, you get your full sequence here. So if you look into the steps of how do you sequence a longer DNA, and in terms of longer DNA, we're talking now of the genomic DNA then, right? So first what you do is you create a genomic library or split the genomic DNA into smaller pieces, right? So you basically split them into smaller pieces here, the ones that are amenable to Sanger sequencing. Then you sequence these individual fragments. And once you get these sequences, you now try to rearrange them in the order they appear in the original DNA. So basically, you go for what is known as an assembly process and arrange the reads in the order they appear in the genome, right? So this is the problem with genome sequencing, that genome sequencing is really challenging. You want to understand the sequence of nucleotides in a given piece of DNA, which is much longer. And we're talking specifically of the human genome, 3.2 billion base pairs, right? So therefore, the challenge is huge, and the technology you have is something that can give you at one stretch in one reaction, give you only 900 bases. So you have to read the entire 3.2 billion base pairs, 900 base pair at a time, right? And so to simplify the problem, what you do is you create multiple small problems and find individual solutions to each problem, and then all solutions are put up together to finally get the original DNA sequence in the order it appears in the original genome. So now coming back to genome sequencing, what is sequencing? Sequencing essentially is to determine the order of nucleotides in a given piece of DNA here, right? And what is our given piece of DNA here is the genome, or one full complement of genetic material in an organism, right? And as I told you in the previous slide, because of the limitations of technology, genome sequencing was back then a very challenging task. And if you go into the history of the human genome, it took us around 10 years and $3.2 billion to sequence the first human genome. And I've already showed you a glimpse why genome sequencing is challenging, given the technology that is Sanger sequencing that was available to us. Now, the question is, why do we want to sequence genomes? Well, we want to sequence genomes because we first want to understand what is there in the genetic code in terms of what makes us human, what makes a chimpanzee, what are the differences between humans and chimpanzees. We're also interested in identifying variations within the same population. So we're interested in knowing what is the genetic composition of one particular, we are also interested in understanding the variations, the extent of variations that are there in the human genome. So when two individuals are compared, what are the positions that they can differ and still remain human? We would also want to compare variations between, so we are interested in identifying variations which could be intra-species within the human population. What is the extent of variation which is possible? What is the positions where most commonly variations occur, and so on and so forth. We are also interested in interspecies variations between humans and chimpanzees. How much is the extent of conservation? What are the positions again where there are differences and variations, and to what extent the variations are there? But more importantly, we are concerned with the biomedical applications of identifying the variations. So not all variations are associated with disease. We are specifically interested in identifying mutations that render people susceptible to diseases. And once you are able to identify these positions where mutations can result in susceptibility to a disease, you could go for a genetic engineering, you could go for genetic engineering solutions for the problem. You could go for genetic engineering solutions for the problem, right? So these are some of the reasons why we are interested in genome sequencing.